I just moved to New York City. I arrived actually on Saturday, so it's pretty exciting. I'm very happy to become a part of this community, so thank you for inviting me. So I came here to talk about causal networks and biology, and I hope I don't derive and talk about boxes and moving trucks, but that shouldn't happen. So I would like to share some thoughts and some directions I've been pursuing at this intersection of learning networks and causal networks in particular, and single-cell data perturbation and gene networks. So I will start giving an introduction about drug discovery because I actually spent my postdoc at Genentech, and these are things I've been thinking about. So I want to kind of give two different paradigms for drug discovery and use this as an introduction for talking about why we need networks in biology. So the different ways of potentially making drugs, the first way would be something called phenotypic discovery, in which we basically would have a lot of library of molecules. We would be able to perform some kind of screening and select some molecules that behave in, let's say, the right way. And out of those molecules, then we would have candidates for making a drug. And here, what's important to remember is that what matters is somehow the what. We don't care about how the drug works, we just care about what it does. And a famous example of that is aspirin, for which we still don't exactly know the mechanism, but it just works the way it's supposed to. By opposition, we have something called target-based discovery, in which we are able to get into the biology to map gene networks of understanding this gene regulates that other one. And based on that, we can select where in the network to kind of intervene with a drug. We call that, I guess, that's the target part. We can validate those targets with different type of models and then make a drug. So here what matters is somehow the how. And we call that the mechanism in biology, but it could be basically the network. And it turns out there is a whole area of biology that could be called causal biology. That's basically the bread and butter of target discovery. And for this, there is a lot of different tools of genetic engineering that help us find those targets. So for example, one type of assay that or experiments that has been available for already almost 10 years helps us using CRISPR, which are genetic engineering tools called genetic scissors, allow us to change the DNA in cells and measure. So by changing the DNA, we can cut genes, for example, or disable them, and we can see how activating or inhibiting a certain gene will change expression of other genes. And that's already helping us with a few things. For example, understanding which genes regulate which other gene, so mapping somehow some aspect of the network. If we can map the full network, then we can also understand how are those relationships between genes mediated. And ideally, once we have a full network, we can kind of understand if we have some perturbations we haven't done yet in our data, predicting the effect of those unmeasured perturbations. So turns out there is something that's called causal machine learning, and it's not because they share the same name that they're necessarily related. And it's still, I still think that there are quite interesting ideas that can be used for the purpose of doing this causal biology. So I'm going to present two paradigms very quickly. One of them is causal structure learning, in which we basically want to fit graph to those gene expression data. So in this graph, every node will be a gene, and the edges indicate some kind of causal relationships between those variables. And we can observe data either from something called observational data, meaning we just let the system unperturbed. When we see how the cells are, our gene expression is correlated, for example, and try to use that for gaining some structure. Or we can also use the interventions with CRISPR to kind of knocking down a gene, and we observe the interventional data. And when we, in this classical setting of causal structural learning, there are many ways of modeling those interventions, those perturbations. One of them is that it breaks dependency. So here we intervene on gene D, and we break dependencies from its parents. So let's say the gene expression would be 0 if we deactivate the gene or knock it out. So another kind of framework is called causal representation learning, in which we reason not at the level of gene expression. So here we have additional nodes in our network. Those can be latent variables, and we somehow model the interventions at the level of the latent variables. It sounds harder, and it is harder, but in a sense, it can be interesting to think about this in the context of learning biology at different resolutions and thinking about what resolution is needed to solve which problem. So the bummer part of this talk is I believe that the impact of those causal machine learning methods on single-cell data or on perturbation biology has been delayed. I have two sets of, I guess, evidence for that. The first one is that there are many machine learning papers describing those methods with some application, and I wrote some of them, and it's really hard to actually write a biology paper in which you have new data and you apply those methods. So I've broken my teeth a few times trying to do that. The reason I believe is that the causal graphs are actually very hard to interpret. So you can always run an algorithm, you can tailor your algorithm, you can come up with some metrics and think that you are building better methods, but when you apply them to real data, you need to collaborate with a biologist, and you need to find evidence that your graph is better than other graphs. And somehow it is actually very challenging in that specific setting, again, because you end up with graphs of thousands of nodes, or tens of thousands of nodes and hundreds of thousands of edges, and you need to figure out which one to prioritize for experiments. So there is a lot of downstream work that needs to be done somehow for building a convincing case. And the other important thing is that validating those direct effects you might find with this kind of graph inference method, the validation is inherently difficult because it does require a biochemical assay of learning, every gene is encoded into a protein, and if you have an edge, for example, those proteins interact, and screening for those interactions is actually not super easy to do. Another source of evidence is that actually there are some competitions that involve the benchmarking on this type of data. The task in this competition is not directly related to learning a graph, but it's more of a downstream application. If we were able to learn those graphs well, then we would be able to predict the effect of unseen perturbation decently. And turns out with those competitions, for example, that I highlight here, we've learned that the causal methods don't work as well or nearly as well than just supervised learning by featurizing the genes in a certain way, like with ESM, learning a protein, embedding of the gene and predicting. So these are my arguments for claiming that the impact is delayed. And so now I guess now you understand that, and you're with me in this emotional and intellectual roller coaster. And so I would like to spend the rest of the talk talking about three different limitations, I think, of current work in machine learning and causal, like network learning for biology. Talking about the model assumptions, scalability, and about the data as well. And for each, I will talk about specificity of this problem, why it's relevant, some kind of recent progress, and then opportunities. So this is mainly an overview talk. All right, so the first message is that the assumptions in this network methods are basically too restrictive. And why I say that is because often case in causal structural learning, the goal is to learn a directed acyclic graph. And so there are reasons for that. They have really nice theoretical properties, and we can quantify identifiability, and there is a lot of theory associated with that. In this case, the data generative process would look like each data set is associated with a graph and with a set of parametric or could be non-parametric conditional distributions like this. And basically, the value at every node depends on the value at its parents plus some intrinsic noise. And the key issue with that kind of parameterization is that we know that in biology there are a lot of feedback loops, and we can already see that with the current data. You perturb a gene, you see changes in, let's say, you perturb gene A, you see changes in gene B, but if you perturb gene B, you also see changes in the other gene, which wouldn't happen if you had this acyclic type of graphs. And the theory is also very difficult with cyclical graphs, so that's kind of a whole problem. Another argument that makes me think that the restrictions are the assumptions are a bit restrictive is it's not just about the assumption, it's also about the available toolbox that we have. So if you think about statistical machine learning or statistical inference, we can do a bunch of things, and it's very easy to derive new models and solve problems. With causal machine learning, it's very difficult to do any of the things I mentioned here. So if you want to, here we have just on the left intrinsic noise, but if you want to add observation noise, then you need to kind of have more specific assumptions and more specific machinery, and for every different type of models, you need different type of inference algorithms. It's very custom, and it's very difficult to actually make it work. If you want to add latent variables as well, we don't have a good toolbox or machinery for doing these kind of things. Controlling for unwanted variation, or even proposing different type of interventions and selecting which class of model is better than others, or diagnostic tools, we don't have any of those things here. So there has been some progress especially with respect to the first part. So for example, now there is interest in thinking about causal modeling in the context of diffusions. So the reason for which it's appealing is that we don't need to have a directed acyclic graph anymore, because we just need to learn those functions, f and sigma here for the diffusion. The big problem is that when we do that, we kind of drift away from any graph at all. And so it might not be, you could still claim that you can post-process these kind of functions to get a graph. But yeah, that's kind of a compromise that right now we have to make. We've done some work as well in this area by not using a diffusion, but using flows. And so using those contractive residual flows, we were able to learn cyclical causal models in that sense. And there is some work also going into latent space and learning directed acyclic graphs in latent space. And it's kind of interesting. I believe that if a process is cyclical and you embed it into a high-dimensional space, you probably still cannot represent a lot of cyclical behavior. So I'm not sure entirely how this is. Let's say that this is a great direction to think about causal representation learning in this setting, but I'm not sure if this embedding part is actually going to solve, for example, acyclicity problems or other relevance problem for biology. So in terms of opportunities, I think we should think about what are general diagnosis tools for causal structural learning, thinking about model selection procedure, especially for selecting intervention models, and globally more like general inferential frameworks for cyclical causal learning. My second message is about scalability, and that it's difficult with this network learning methods to basically get them to scale. There are basic, for those of you who aren't familiar with those methods, there are two key reasons. The first one is that when you write a structural equation models, you basically, if you take this function f here and you put a linear map, you see that for solving this system, you need to invert a linear system. And so, except if it's a directed acyclic graph, you will need to pay the price of the matrix inversion. And if it's a DAG, you need to then do your optimization in the space of DAGs. And that's very tedious. And it's at least also cubic somehow to deal with those, either a score or to work in that space. So this is a root of the scalability issue. And there are a few methods that try to circumvent this problem, but they have their own issues. So for example, I've mentioned diffusions before, and we know that diffusion scale also has huge scaling issue with respect to the dimension of the problem. And also the optimization problem of, if for causal representation learning, we want to learn the map, that takes you from latent to observed data. And also, we need to learn the graph. And that's very challenging to learn this at the same time. So there has been some progress in that area, basically thinking about numerical stability of some of those scoring functions for DAGs. And Achille Nazare, this student at Columbia, has worked on studying the numerical stability when the size of the graph becomes huge, and making those algorithms more stable for differential causal discovery. I thought that was pretty interesting. I've done some work in trying to put hard constraints onto those models to basically encode, to factorize them. So instead of learning gene-to-gene relationships, trying to learn relationships that are mediated through factors, and that corresponds to learning a bipartite graph. And here the factors are basically gene programs or gene modules, and you can attach a score to them that correspond to the activity of that gene program, which is a nice biological hypothesis because we may not be able to recover all the causal relationship between genes, but potentially between different molecular processes, so at a higher resolution. And then there are also some really interesting work around causal discovery foundation models that basically work by collecting statistics of conditional independence across different subsampling of nodes and collating them together with a transformer. And so I think that could be very helpful for solving those huge causal structural learning problems. So I think there is an opportunity here to think about learning across multiple resolutions. This idea that a given data set might tell you some biology, but only at some specific level, and you will not be able to resolve causal dependencies between all the molecules, or learning a causal graph between every pair of molecules is, I think, very important. And we don't really have any identifiability guarantee, or we don't have any theoretical guarantees, I guess, that tell you whether there is a single resolution at which you would be learning, for example. I think that I'm quite interested in thinking about those problems. There is also something interesting to think about in the context of we need to solve this. So there is not a single network of biology. The network is very dependent upon context. So for different cell types, you will have slightly different networks. And so trying to have algorithms that can tell you which part of the network is different across different cell types, or being able to recycle information across different contexts will be very interesting as well. And finally, my last message is about the fact that the available data does not tell the whole story. So I think as a computational biologist, it's a great message to say, because we can always blame it on the data generation part. But it's actually very important also as computational biologists, to own the data that we have and be able to understand what we can draw and what we cannot draw from it as conclusions. So here is a question to which we all know the answer. But it's true that we cannot expect a model to infer things that's not in the data at all. And somehow it's difficult in biology, especially because we don't always know what the data tell us. So it requires extra thoughts. I want to present two different types of points to illustrate that. The first one is that in biology, I guess we're in this specific case of gene expression measurements. We only measure gene expression. There are so many things we don't measure. For example, so here in our structural causal model, we basically want to put an edge between two genes. Let's say here a transcription factor. A transcription factor will be one of those genes that once encoded translated into a protein will be able to bind to the DNA and regulate another gene. So that's why it's convenient as an example. So if we want to put an edge between a given transcription factor and one of those target genes, what's actually happening is that, again, the transcription factor is translated into a protein. The protein needs to bind to the DNA. And then something happens at the DNA level and will regulate expression of another gene that's called the target gene. But so many of those steps could be actually dependent on the expression of like a third gene or another transcription factor or phosphorylation. So there is just a lot of intermediate steps that are actually completely removed from our mental model or from our computational model. Another kind of missing information is about temporality. So when we look at those screens, and when we look at how the DAG, directed acyclic graph or structural causal model work, we basically think that the changes from TF to target gene is kind of instantaneous. But what's actually happening is that cells are dynamical systems. And so when we do the experiment, we need to wait a given amount of time before measuring gene expression. And if we wait too long, some of the genes return to baseline, and some of the genes are still changing. And so we were not able to kind of use that temporality for all the causality. And so there could be a lot of different networks explaining the same data. And having more temporal information could help us recapitulate more of the causality somehow. And so I want to just highlight a few recent progress here, and that's mainly because there is extra data that's helping us figure out some of these problems, but the data is emerging and very new. So one idea is if you don't observe enough things, you can always observe more things. I don't think that's a key solution to the problem, but it definitely paves the way and opens up new way of having added resolution. So what we're able now is with perturbations to also measure gene expression and chromatin. So in this example, this specific paper, there are no perturbation, but they're using some pseudo-time as a way of controlling for variation like this. And what it helps with is you can track at which point in time the chromatin is opening, and then you can point at, understand at which later time point the RNA of the gene expression of a gene nearby is also changing. And by being able to, like this, capture those correlation across time, you're more susceptible for picking out putative causal relationships between genes, for example. And so we could do this with perturbations as well, etc. And in terms of time, yeah, this is on the right is a model that I've been working on, where we basically use flow matching for fitting an ordinary differential equation to this temporal data from perturbations. So on the right, you can see every color is a different perturbation, let's say a chemical, and you observe at different time point the gene expression. And the data sets get pretty large because you end up with 1.5 million cells and gene expression for all of those cells, and around 300 different drugs. And you can you aim to track like this, like what are the trajectories of how genes go up and down through time, and then learning causal relationships, I guess, between those genes with that kind of data. And so the additional opportunities, I guess I've talked through most of them already, is if we can, by having new modalities, we're able to add a new semantic of biology, because now we can look at the opening and closing of the DNA, for example. We can look at different proteins as well. And it helps to reason across those modalities for figuring out new biology. One thing that I haven't mentioned, and I think is quite important, is given that we have indeterminacy problems, that there could be many graphs explaining the same data, it would be great to find a way to softly incorporate prior knowledge, such that we're basically shrinking out of all the possible graphs that are equally explain the data, having a way of shrinking to those that are biologically plausible would be very important. I'm also interested in experimental design in my research in general, and thinking about how by acquiring more data, we would be able to resolve some additional indeterminacies. And finally, something that I'm really excited about is to keep thinking about this intersection of causal network, causal machine learning, and dynamical systems. And I think that, this is something that I'm quite passionate about. All right, so as a conclusion, we started this journey with high expectations for causal machine learning in molecular biology. We found that there are quite a few bottlenecks, and the idea is that there is a huge, I think there is a huge discrepancy between the current, the theoretical developments and the features that are needed to solve those biological problems, basically. And I think that finding this kind of balance between, the theory is a theory, and it's always interesting in its own. and somehow the development of algorithms could maybe benefit from having a different view on like what's needed for some of those scientific problems. And what I'm interested in working on is having cross-disciplinary collaborations between machine learning researchers and biologists to kind of enable building those new tools and discover new biology on these novel data sets that are emerging. Thank you very much. Yes.
You mentioned manually adding latent variables to the existing graph or the network.
Yes.
Why doing that? Like, what downstream task do you have in mind that knowing that would help?
Yeah, so latent variables could be helpful in the context of, again, maybe in the context of resolution. So maybe you don't need to know exactly which gene regulates which other gene, but you care about, for example, summarizing plenty of genes into different buckets that have the same function, and then knowing which function, which pathway regulates which other pathways. And so whenever you think about this like extra higher level view on the data, those are latent variables. So you need to infer them. And then when you start writing mathematically like structural equation models with latent variables and you need to infer those variables and learn the causal graph at the same time, that's finicky. So that was my point.